Color your life with you so bright. Paint every moment with all your might. Believe in yourself, let hope ignite. A symphony of strength, a beautiful sight.
Okay, I'm very happy to say we do have Michele, who with us. She's joined us about 5 minutes ago. A little worried that she wouldn't make it, but she's always there. She's always been there for me over my 20, 15, 16, 17, 18 years of Parkinson's. I've lost count. Must be a sign I have Parkinson's. Um, she's a big supporter of our centre too in the European Parkinson Therapy Centre. Okay, I'm very happy to say we do have Michele, who with us. She's joined us about 5 minutes ago. A little worried that she wouldn't make it, but she's always there. She's always been there for me over my 20, 15, 16, 17, 18 years of Parkinson's. I've lost count. Must be a sign I have Parkinson's. Um, she's a big supporter of our centre too in the European Parkinson Therapy Centre.
Thank you, Alex. It's lovely to be here. I'm always going to be here if you ask me. I won't let you down. So just to introduce myself, I've been a neurologist focusing on Parkinson's and movement disorders for 20 years in the NHS. I'm based in Oxford University Hospitals. I'm also Professor of Clinical Neuroscience when I transitioned from working in the coalface of NHS clinics in the UK to doing a mixture. And so I still do Parkinson's clinics every week, but I also do research. And I'm going to tell you a little bit about my research and how I hope it's going to help deliver better treatments and clinical trials for people with Parkinson's. So I'm going to share my screen. Had you seen this slide yet with the 4 boxes?
No.
Okay, so this just summarizes what I've been saying about the cohort, the discovery cohort. And this is like when you go around the world, when we first set this up in 2010, nobody knew who we were. And then you have to kind of gather data, and there's about a 10-year period of really hard work where it just sort of feels like you're plugging away, but there's not a lot of results. And then all of a sudden, everybody wants to work with you and collaborate, and because actually you've got some really interesting data. And this is all related to the Parkinson's patients who kindly volunteered their time. So we have over 1,800 people in this cohort, and we've had quite a lot of over 200 papers published from this data. And we also share our data to other researchers and scientific collaborators. So talking firstly about the Parkinson cohort, this is the map of where we recruited our patients. And right in the middle at number 1 is Oxford. And then this whole area I'm calling the Thames Valley. All of the red dots are district general hospitals that are linked to Oxford. And you can basically, on a good day, drive. I mean, Kettering and Northampton are a bit longer than an hour. The whole population we cover here is 2.4 million. So we recruited people within 3.5 years of their Parkinson diagnosis from 2010 to 15. And we recruited 1,000 people. And then we also recruited now 312 people from 3 sleep clinics in the UK. And here I'm showing you the doctors that helped to go around the country to Sheffield, Papworth, which is now in Cambridge, Edinburgh and Oxford. And what you can see here is that we've had 21% of our Parkinson patients of our RBD have converted already to Parkinson's or dementia with Lewy body or MSA. And the main thing about these conditions is that they're all alpha-synuclein disorders. So they're all related to misfolding of the alpha-synuclein protein in the brain. And what I'm going to show you here is data from Oxford Discovery, which were nearly 1,000 people with early Parkinson's. And I'm also going to combine that data to 1,601 from Discovery. And here you see Donald Grosset, who was the cohort lead for something called Tracking Parkinson's. So Tracking Parkinson's is also funded by Parkinson's UK, and it has a very similar way of assessing people, the same protocol. And so what it allows us to do is to test our hypotheses or find a research discovery in one cohort and then see if it replicates in a totally separate one. And when you've got that ability to do these two independent cohorts and then combine the data, it's very powerful, because you've got between you over 2,545 people, all diagnosed with Parkinson's in the last 3 years, and they've been followed up every 18 months for an average of around 10 years. The people in Bristol are Professor Ben Shlomo and Michael Lawton, who are the statisticians who helped with this research and combining the cohorts. So this is just showing you the table that, as you see with all cohorts, roughly male to female is more common by about 2 to 1 for Parkinson's. The mean age of diagnosis is 66 years, and most people, the mean had only 1.2 years of Parkinson's symptoms from diagnosis when they were first seen, and only around 10% when they were first seen didn't have any Parkinson's treatment. So they were what we call drug naive or untreated. Now, this bullseye illustrates what we were able to show, which are 4 independent clusters or patient phenotypes. And the way that we did this was to put all the data that we collect in our 2,000 patients that were clinical data. So it would include things like your age, your gender, your date of diagnosis, your clinical examination, your rating scales, your memory, and your memory function, and your non-motor function, which includes constipation, severity, sleep problems, loss of sense of smell, depression, anxiety, and memory. And when we put all this data in, firstly on the tracking cohort that I've shown you, and then validated that in the discovery cohort, just using the clinical information at baseline, so that's within 1 year of your diagnosis, we were able to discern 4 separate clusters of patients. So I'm going to go through and describe them now. Basically, black is bad, and white or pale grey is good or better than average. So if you start in the middle bullseye, it's about motor progression. So it's how fast or slow you progress in the next 3 years after your visit. So here you can see that cluster 1 has the fastest motor progression, but cluster 4 the slowest. The next ring out is your levodopa benefit, and that was measured on a levodopa challenge test. So you've got poor response as darker grey and good response as pale. The next is your baseline non-motor. So that's your sense of smell, sleep, depression, anxiety, memory, blood pressure drop at your baseline. And lastly, the outer ring, I hope you can see my mouse, is your baseline motor symptoms. What you can see is that cluster 4, for example, has the most unilateral tremor dominant motor symptoms. So that's what we call tremor dominant Parkinson's. And we've always recognized that there is this subgroup when you do a Parkinson clinic. These patients have average baseline non-motor. They don't tend to respond so well to Parkinson's levodopa medication, but they progress slowly and they generally do quite well. You can see that cluster 2 on the opposite side is that group of people who are really mild. They have a good response to medication, which it means that they tend to do really well, and they keep coming back to clinic and they don't seem to be getting very worse. And they say, oh, my friends are getting much worse, but I'm really good. Cluster 3 and cluster 1 have a lot of black, meaning that their baseline non-motor symptoms, in particular for cluster 3, and their baseline motor symptoms are much worse, but also they have only an intermediate or poor benefit with levodopa medications. So what we then did is we looked at the baseline clinical subtype, and then we fitted what happens in both the tracking Parkinson's cohort and the discovery cohort to the motor progression over the next 3 years. And the main message here is that this replicates very well across both cohorts. So for the tracking Parkinson cohort, cluster 3 has the highest motor problem at baseline at 32, and it's around 30 for discovery, but also progression across cluster 1 and cluster 4, if you just look at the difference, is very meaningful.
Wow.
Because the difference is 3 motor UPDRS points per year. What that is a clinical endpoint in many clinical trials. So what I'm arguing here is that if you do not account at baseline trial recruitment for these differences in your patient group and how they're going to progress and how they present, you could have a false positive or a false negative clinical trial result. It's not the fault of the drug, but it's our fault because we haven't understood patient phenotype or cluster. We haven't adjusted for that in the trial. There's some really interesting evidence that's been published on prasinezumab, which is a intravenous therapy. It's a monoclonal antibody to alpha-synuclein, and that was an overall negative trial between Parkinson's given the drug and Parkinson's on placebo. But a sub-analysis later showed that the people who had the fastest progressing Parkinson's benefited significantly, but not the others. So these differences are here. We've known it as long as people have looked after people with Parkinson's. But at the moment, and when we do a trial, we just recruit you if you've got Parkinson's within how many years. And then this next one is showing the average change in your memory or cognitive function. And again, we see that there are quite big differences which are replicated across these two cohorts in terms of fast versus slow cognitive progressors. So what I've spent the next 5 years doing is looking to see if there's any very simple lab measures or biochemical measures or genetic differences across these 4 clusters. And this is a summary of probably about 6 years of work. And in essence, cluster 3, which is the worst one at baseline, that progresses the fastest in terms of memory, has the highest rate of GBA mutations. It's around 10% compared to only 2% in cluster 2. And that makes sense because we know that people who have Parkinson's and a GBA mutation progress more quickly, and they are more likely to get dementia earlier, and they get diagnosis of Parkinson's 10 years earlier. The other thing is that this cluster has pro-inflammatory response on their blood tests. So that suggests that an anti-inflammatory medication or treatment might be very important for this cluster, but not very important for cluster 2, which has the opposite, and it has no evidence of an inflammatory response on basic blood tests. This cluster 3 at baseline also has a vascular risk because they have reduced HDL cholesterol, which is the good cholesterol. And they also have, as I've said, the highest rate of cognitive progression. Cluster 4, which is this tremor dominant group, have the slowest rate of motor progression, and cluster 2 have the opposite, the lowest rates of GBA. So how can we use digital tools to try and understand this heterogeneity, this individual difference, both in how you present and progress from your Parkinson's? And why would you want to do it? Well, one of the advantages is that it gives you real world data on how that individual is doing at home over a longer period of time. And that's really important because whenever I see people in clinic, they always do their best walk, they swing their arms, they stand really straight, and they don't have freezing. And the partner is usually like, he never does that at home. He's constantly falling over or freezing. And like, here he is striding down the corridor. So real world data, it allows you to collect and measure changes over very shorter periods of time because you're getting more data. And that could be days, weeks, and months. I think the data we collect at home has greater granularity than our clinical scales that are currently gold standard. Could digital measures be really important because they reduce placebo effect in Parkinson's? Now we know that in Parkinson's, the placebo effect is very strong. I'm going to talk to that relevant to the exenatide PD3 trial in a moment. And it may be from this study showing here in Huntington's disease, where the digital measure, which involves gripping a thing and measuring the pressure, and that didn't require the doctor to measure it, but it might have been a research nurse, versus the clinician rating scale for Huntington's, that clinician scale showed a big placebo effect because people who thought they were on the drug actually improved over time, but people who were doing the digital measure had no improvement. So that's really interesting. And if we can demonstrate the same in Parkinson's, I think that's really important, because one of the big problems in clinical trials is this placebo effect. The second is that there is less variety or variation or variability in digital measures compared to when you use a unified Parkinson's rating scale. Why is that? That's because we're human, and you know, a patient might only come every 6 months or a year, so their mental state, how they've slept the night before, their journey to hospital, their parking experience, their stress level have a massive impact in that half hour, 1 hour that they're with the neurologist doing the unified Parkinson's rating scale. So if you can have greater numbers of measures over a period of time, like 7 days, that is bound to be a more accurate reflection of where your Parkinson's is. The challenges of these digital measures are there's way too many of them, and it's understanding what each one does and how valid it is. And usually it's only been tested on very small numbers of patients and not over longer periods of time, so to see how good it is at tracking change. For us as doctors, one of the big issues is regulatory approval, because if we want to use a digital device in a clinical trial, it needs FDA or European Medicines approval, it needs good scientific evidence, and it has to be able to be deployed at scale, it has to be good for the patient to use, and they have to be demonstrated of high compliance, and it does increase cost compared to rating. So one of the things I've been interested to do is to look at the participant user experience of these digitals. And here I'm only going to talk about motor symptoms, and I'm going to think about patient-reported outcome measures. So these are things that are meaningful for patients and their families. First thing to explain, there are two types of digital measures, broadly speaking, and the tech that you use is very similar. So don't be put off by that. Active versus passive. So in active testing, the individual is being asked to do a series of tests at a particular time or frequency in the day or week of their monitoring. So an example of an active test would be the APDM gait measure you can see what this lady is wearing. She's got sensors on her feet, abdomen, you know, wrist watches, and she's being asked to walk 10 meters, turn and come back while counting backwards or not. This is an example of one of our research participants doing the active smartphone test, which I'm going to show you. And these are examples of passive testing. So this device called Activity is very small, and we've been able to sellotape it literally using gauze onto a patient's lumbar back. They go home and it records how much walking, rolling over and turning they do over the next 7 days. Another example of passive testing is a house where people live in that can be rigged up with sensors that detect how much they're walking, going out and moving around in their own home. So in exenatide PD3, which you will all know has sadly been a negative trial, we were funded by Cure Parkinson's Trust to do a exploratory digital project. And I'm going to tell you about this results. So what we were doing and what we were paid to do is to do the smartphone test and to ask participants to take a phone home from clinic and do this test 2 to 3 times a day over 7 days at different time points every 6 months in this 2-year trial. So it would happen at baseline, 6 months, 12 months, 18 months, and 24 months. So we've got this 7-day monitoring with the smartphone and the activity device on the lumbar spine. And we also did some surveys of what did the participant, the patient and their clinician think of using these tests at the beginning and end. So this is the survey. It's very simple. The smartphone survey, do you own a smartphone? Yes, no. How often do you use an app? So we had people who didn't have a smartphone, didn't have Wi-Fi, never used an app, who were able to learn to use this smartphone because people in clinic went through it with them, and we have written instructions. And then before using it, how easy did you think it would be to use? Now that you've used it, how easy was it to use the smartphone app? How willing would you be in future to use this app with your nurse or doctor? And how willing would you be to use the app at home to monitor your Parkinson's outside a clinic? Then we had a similar one for the lumbar back sensor. So the take-home message of the survey is that although we had nearly 400 patients recruited, sorry, it was 200 patients recruited. These are the time points to the overall study. We achieved nearly half of them who said, I'm willing to do the digital smartphone test and the gait. And when we did the survey for the smartphone, 86% of people at the end of the study had managed to do at least one daily recording over the 7 days, which is really important. They found it easy to use, and 96% were happy to use it at in-clinic visits and 89% at home. And for the activity device, between 71 to 75% of participants found it easy to use. In actual fact, they didn't have to use it because it's a passive test, but occasionally it fell off. You could still wear it in the shower, so it's perhaps a little bit more uncomfortable wearing it at night. But that's the take home. We then set up some focus groups, and we then had a spin-out company which was set up by our university support using this app. And here are some of the patient reports using the app. You know, he's using it on his own phone now, so you can download it onto your iPhone or Android. The app was easy to install. The app, having the exercise available on a single device that I carry around with me anyway, makes it very convenient to use. The exercises can be performed anywhere, that you can find a flat surface and a few meters to walk. And, you know, pretty positive feedback from large numbers of patient focus groups, you can see. Based on that, the company is now testing this in 6 Parkinson's NHS clinics and trying to gather data to show that it is effective for patient self-management and for the clinician to make treatment decisions for Parkinson's. So one of the kind of positive notes of the exenatide trial is that we were able to show something really important. And this work was done by Timothy Allberg, who's our postdoctoral researcher. So here is the main result of the exenatide PD3 trial. The primary endpoint, which was the UPDRS Part 3 off score, didn't change over 2 years between the placebo in blue and the exenatide group in red. Okay, so we then also looked at this with our smartphone, and I'm just going to run the video this takes about 7 minutes to complete. You're asked to start with a voice test where you just go ah into the phone, and then you'll see the other tasks. So here he's just telling it he's going to start, and he's standing, and within each smartphone is a tiny bit of kit called an IMU, inertial measurement unit, and it measures how much you're moving in a 3D plane. So he's now putting the phone in his pocket, and he's just walking about 10 meters or at home in a corridor, and he's turning and coming back.
You hope.
And this is a dexterity or a finger tapping test. So what he's being asked to do is to just tap with his fingers in his right hand and then his left hand in a regular rhythm. Okay. And if you have Parkinson's, this regular rhythm gets interrupted, and a bit like freezing, fingers can freeze or slow down. This is the reaction task. So he's asked to press his finger on the screen as soon as he sees a target. The target comes and goes randomly. And then lastly, the really clever bit is we can measure tremor, both rest tremor when the patient has the phone in their hand, resting and is counting backwards, 100, 99, 98. And what you can see here is that this counting backwards, when he closes his eyes, the tremor in the right hand starts. So we're able to measure rest tremor in right and left, but we're also able to measure what we call postural tremor, which people get when they're reaching for items like a pint glass or a cigarette. Not that you parkies do this much. Okay, so that gives you an idea. So what I thought we might try and do, given that the trial was negative, and was see whether we could use the smartphone to predict if you were going to be a fast or a slow progressor in your motor function at the beginning of the trial, at your baseline visit. And if you look, you can see that roughly 1 in 4 of all of the exenatide trial participants had faster motor progression in red, compared to about 3 in 4 that had a slower progression. Can you see the differences here? So this is the score, this is over time, and this is the 2-year. What we then did was say, well, if we just take one smartphone test at baseline, can we use the 1,000 features to predict if you're going to be a fast or slow progressor over the next 2 years? And the answer is yes. It's not 100% accurate. It's about 80% accurate, which is pretty good. And what we then also did was look to see if any of our smartphone measures were different between the treatment exenatide group and the dummy, and we didn't see any differences either, like very consistent with the UPDRS. And interestingly, there are people who were placebo given the dummy drug who got better over the 2 years of their exenatide trial. They didn't just get less worse, they got better. So that's a mystery. Is that a placebo effect? Is that because they're seeing a research team regularly, they're learning more about their health, their Parkinson's, and maybe their mood is better, they're getting their blood pressure checked? Who knows, right? So all these things have a potential. So I hope what I've done now is just briefly show you how digital technology has a potential to personalize your treatment, predict your progression, and also ultimately deliver better clinical trials for Parkinson's. And this is some of my research group who all work incredibly hard. And I'd like to just thank our funders, Parkinson's UK, Michael J. Fox Foundation, NIHR, and Cure Parkinson's Trust. So very happy to be able to take questions.
Okay. I just want to thank you, Michele, because what you showed us was a real insight into a clinical trial and the way it's conducted and the testing levels and the experiences which you have. Now, I've always said that people who have tremor have a milder form of Parkinson's.
Yeah, you're right.
I was right.
You just had that proven in a data-driven AI approach.
Well, ironically, I think people with tremor actually feel that they have a worse form of Parkinson. Because...
And the reason is it's visible to everyone. So it's more socially embarrassing. And when people get anxious, the tremor gets worse immediately and they can't hide it.
Sure. Okay. Well, we're going to open up to anyone else who would like to ask a question. I should just ask you, do you know how many, everyone with Parkinson had a different form of Parkinson, even if they're in 4 specific
broad
cohorts?
clusters. Yeah.
Do you have you actually got a number of the number of types of Parkinson's you've measured?
Yeah. So I mean, the problem is there's lots of different ways of skinning a cat, right? So this sort of idea of patient clustering or data-driven has been applied and done in so many different ways, and there's no real right or wrong way. And effectively, if the data that you put in is good, what you get out is generally also going to be good. But many studies, they don't have these numbers. They don't have the replication of the 2 cohorts I've shown you, both tracking PD and discovery. But if I broadly explain from that, I'll just show you on what we did. The data fitted into a 4-cluster model, but actually you could make it fit just into a 2-cluster, fast or slow motor progressors, for example. And it really depends on the research question you're trying to ask. So if I just share my screen briefly, I will just hopefully be able to get to this. So here what I've done is in percentage terms, I've given you the percentage of people assigned to each of the 4 clusters. And you can see that it varies between 20%, i.e. 1 in 5 of our 3,500 people with Parkinson's had tremor-dominant Parkinson's, up to 21% for the worst baseline cluster 3, and 31% for cluster 1, 29% for cluster 2. So it's roughly, quite equally distributed.
regarding clinical Parkinson's or research that may be done, or the cluster group she was showing. I think some people with tremor may disagree with her assessment. And if you would like to just raise your hand or just take off your mute and say hello. If anybody would like to do that. Lisa. No?
Hello, can I have something, please? Paul Brown here.
Yeah.
Hello. On this trial you mentioned, and very many others that I've seen, there's very little notice taken of the timing of medication. So I know that medication is very effective for me, and if I do one of these tests within, say, an hour taking some medication, dopamine, L-dopa, then I'll do very well. I wait another couple of hours, I'll do very badly. I mean, somehow that medication timing must be taken into account, surely.
Absolutely, it is. So you've raised a really important point here. And for the exenatide trial, the main outcome measure was a change in the MDS-UPDRS when the patient had undergone an overnight withdrawal of their Parkinson medication. So they were in off phase and they were examined in the morning. So when I showed you the trial thingy, their patients had to undergo this medication withdrawal overnight 5 times in the trial, right? Baseline 6 months, 12 months, 18 and 24 months. And it's the off medication data that I showed you that was negative, as was the on trial data over the 2 years in the placebo versus exenatide group. But the smartphone could actually predict fast or slow progression at baseline from both the on or the off. So the good news for that is for patients who really don't like going off, which I completely understand, you could still use the smartphone to predict progression as accurately, and the on is just as good as the off for doing that. So, what we've done for a lot of trials is make the patient withdraw from medication overnight to get this off assessment. But I'm not sure how practical it is, especially when you're, asking patients who've lived with their Parkinson's for sort of more than 5 or 10 years. So they're fluctuating anyway. They say it can take days before they get back to how they were before. It can often be quite anxiety inducing to be off meds and helpless or feel that you are. And I think that the field is now moving to doing on medication for many trials and doing it remotely, either with digital or patient questionnaire.
We've got a question coming in from the chat, which is from Graham Jackson. Could the smartphone be used to identify when a change or increase in medication is necessary?
Yeah. So that's a great question. I'm so glad. It's really great. You've got a very intelligent audience.
You planted these questions, didn't you?
No. So that's exactly what the New Health is trying to do. Because basically what I'm doing is if I see someone in clinic and I've just made that diagnosis, I'm saying to them, would you be interested in trialing this free smartphone app on your own phone? And I ask them to do a week of baseline recording before they start treatment. And then I say, take your pills and then continue to do your recordings over the next 6 to 12 months. And then that patient's results are fed back to them over time. So they rank their symptoms. What bothers you the most? Motor, is it tremor? Is it stiffness? Is it slowness? And then the features of the tremor scores, the stiffness, the mobility are fed back to them so they can see if they're getting better or worse with the medication. And also it allows you to look to see how much benefit you get with medication, which is a really important clinical factor for deciding if you might benefit later on for deep brain stimulation or complex therapies. And it could also allow the clinician, so I get access to it when they come back to clinic, to say, look, I think you might need an increase in your medication, or I can see you're fluctuating a lot in the day. Maybe we need to think about complex therapies. So the only market available alternative that I'm aware of this currently in the UK is called the PKG device, which is a wrist logger that the patient wears over 7 days. Now that is quite expensive. I'm not going to mention the cost, but my NHS trust said they can't afford to pay per report of the PKG device. So it is available in some hospitals, but the smartphone, you can use your own smartphone and it's pretty cheap and it's all automated. So the results come back to you in real time. So absolutely, I think it could be used in future to actually adjust your treatment.
Okay. Anyone else would like to ask a question? Otherwise, I will ask a question. Okay. No one? There's so many of you that I can't believe none of you are actually going to actually ask anything.
Everybody's shy.
That's you terrify us. You would terrified me.
I will say I did do an NIHR webinar, and it was chaired by Rory Ketland-Jones. And after the hour, I was exhausted. I had non-stop questions. I reckon I had probably at least 30 questions in the hour. And
I'm
then
going to
Feeney
tell
wants to
you.
ask a question. Mary Feeney is trying to put her hand up.
Okay. Mary.
She's not muted. She's not.
Okay. Isabel.
I live in Canada. Is the same sort of trials available over here? Do you know of any?
The honest answer is I work with only one Parkinson neurologist in Canada called Ron Postuma, who's based in Montreal. But the exenatide trial, I'm not aware of a phase 3 trial that's been done in Parkinson's other than this UK one. But quite a lot of the trials, for example, I know for a fact that the antibody trial called prasinezumab with Roche has been done in Montreal. And in some parts of the world, for example, in Australia, the Australian government is funding Professor Simon Lewis to do what's called a multi-arm, multi-study trial, where they're testing 3 drugs to see if they slow down progression against 1 placebo. And in the UK, we're also going to be starting that. It's called ACTPD. We're starting that in the next few months. So I think there is availability. What I would say is you must have a national Parkinson's charity for Canada.
We do.
They often have what's called a trial finder website, or you can email. And we do this in the UK. So you can actually put in your address and it will signpost you to the nearest studies and centres.
Right. I think there's a new one.
Also have the trial finder, which is global.
Right.
Okay.
You could do the Fox trial finder.
Thank you.
I've got to say that there's an interesting question just coming on chat, slightly off topic. But do you think you can alter progression of Parkinson's using your mind, psychology?
Very interesting. Okay, so let me give you an example. If you've got really bad anxiety, and we definitely look after many patients, often younger when they get their Parkinson's, and they've got anxiety predating their diagnosis by 10 years, and it's a really prominent feature for their Parkinson's. I think it has so many negative impacts that treating the anxiety helps your motor symptoms. Many people with anxiety, for example, don't sleep well. They're chronic insomniacs. It's also a problem for many people with Parkinson's. I would say that if you can improve the anxiety and you improve the sleep, you're actually going to improve sleep benefit. What's sleep benefit? That's what a proportion of people with Parkinson's get, where they feel much better in the morning following a sleep before they've had any of their medication. And it doesn't happen for everyone, but it's a very interesting phenomenon. We also know that if you disrupt somebody's sleep, your memory and your cognitive function is worse. And there may be some protective brain mechanisms that happen only when you're asleep. For example, clearance of misfolded alpha-synuclein and other proteins happens only in deep, slow wave sleep, and that's called lymphatic clearance. So, what I'm really interested in is if you treated poor sleep or anxiety symptoms, would you actually alter the disease trajectory of an individual? And it's something that we need to do a trial and test, because I think it's very likely, just from looking after patients over the years, I've seen the effect that it has.
I think positive thinking had a major role to play. I honestly do.
The other thing
Um,
is that if you can engage with the patient at diagnosis and tell them about research trials, they do better. So general health and Parkinson's does better. The more you know, the more you can fight it. The second thing is that if you can tell someone that exercise is the only proven treatment that slows down progression, my experience is if people really embrace that, they do so much better over 20 years with regular high intensity exercise.
okay,
Do
Mary.
I have research to back up the exercise? I have at least 4 phase 2 or phase 3 clinical trials of exercise that have been published. The problem with exercise is getting a placebo group that haven't had exercise but have had stretching or handwriting. So how do you deal with that effect? And the second issue is that the treatment trials of exercise are very short, 6 months to a year. So what I want is to get funding to do a longer trial to look at the lifelong effects of exercise. I also saw a thing on the chat. Do UK neurologists know about New Health app? The answer is yes, because 6 UK Parkinson's outpatient clinics and hospitals are using it now, and they're trialing it in people attending clinic for free, specifically to see if it can improve the management of people with Parkinson's.
Okay, Mary Feeney, I believe you've been waiting quietly in the wings.
Well, I have, and a lot of what I was going to talk about or ask about has been covered just now by dealing with the nervous anxiety symptoms and also sleep patterns. My sleep pattern has generally been good, but it has been disturbed when I had a change of medicine. I got terrible nightmares, and I've since diluted. They have me on half time on that particular medication and the rest of the time on Cinnamate, and that has helped. I don't generally have nightmares, and they were horrifying, so I really couldn't stay on that combination. But I am now on a half combination. But what I wanted to mention to you was this: where would you categorize in terms of symptoms toes curling? Because it sounds funny, but it's actually very uncomfortable. Agony. It's agony, and it's hard to get them unstretched. And I do a lot of exercise. I agree wholeheartedly with you. The change, I was diagnosed in 2010, and I was diagnosed with mild tremor. And I didn't progress very much for many years. And that had been predicted by my doctor here in Dublin. And I'm actually in West Cork at the moment, but I live in Dublin. And I want to know why I had tremor. My tremor started, I was tremor dominant. It started on the right side and it's gradually moved to the left side. I used to be able to squash it by putting one foot over the other when I was working and having to talk. So I have pursued every avenue that I could imagine. And I believe wholeheartedly in exercise. I do Pilates, dancing, other people do Irish dancing here, which is supposed to be very good. And in general, I've kept up interest. I like to think that cognitively, I'm okay anyway.
You seem pretty on the ball cognitively. If you can go and attend a Zoom call, and you've told me your history. So, I mean, I think it's quite complicated when a person with Parkinson's gets foot curling or cramping. First, just some very general observations are these are dystonia, right? We call it dystonia, and that's a medical term for an abnormal posture. It can affect the whole body or just one part, like the feet. It can affect the neck or the hand. And it's more common for younger onset people with Parkinson's to have dystonia. What's your definition of young onset? Well, strictly less than 40, But
I tell you, I'm 72 now. I got it when I was 58.
So, you can also include under the age of 60. And then it can be two things. The foot cramping can be when the medication is wearing off. So a lot of people will get that as an end of dose wearing off, or really painful with like neck and, as well. First thing in the morning at 5 or 6 a.m. Many of my young onset patients get that. Before they've actually taken their medication, or when they're taking it before they switch on, they can go through this very painful thing. I think the other thing is you can get foot cramp, irrespective of whether you're on or off medication. So you've always got to just think a bit about, is it an off? Is it an on? Is it a transitional phase? So for you, it's off. The other thing is you have to make sure you're not confusing it with something else like restless leg syndrome, where people get this feeling of cramping or discomfort, they have to move, but the foot itself is not in a spasm. And how to treat it depends on the cause, right? So if it's an off-period problem, then you want to be reducing the off periods, right? But if it's a transitional thing, you want to be kind of smoothing out the PD motor dopaminergic stimulation. Basically, it's not an easy way. You also, I found, what really helps is just having an orthotic review where they can take a mold of your feet and give you insoles, changing your shoes. And sometimes I'll try, in addition to dopaminergic medication, anticholinergic medication, which is specifically there for dystonia. But you have to watch out for potential side effects.
Well, I will investigate that. But I think that it is one of the most serious complications because it prevents you walking easily, and that's critical. I want to be able to continue walking. I have more falls, and I mean, I have 2 hip replacements as well, so that probably hasn't helped. But I recovered from those fully within a year, and the last one was last year.
That's very good. So you need something probably to reduce your off periods.
Yeah.
In a kind of very reproducible way, without compromising by increasing your dyskinesia. So that's a tricky one, isn't it?
I don't want to concentrate on my condition, but I wonder, do other people have this problem?
The foot cramping.
Yeah. Because my toes can be completely straight one minute and completely cramped the next.
Well, maybe people could, we could do a straw poll, put your hand up if you get this problem and you've got Parkinson's. Because I suspect.
I can confirm that.
Yeah, I suspect it's at least, 30 to 50% of people will have had it at some stage.
Yeah, there's quite a lot of hands going up. Yeah.
There you go.
Yes. Thank you very much. That's been very.
Okay.
I'm finding it very
interesting. Thank you.
Thank you. I'd like to do a try.
Thank you, Mary. There's a question whether the app is available in other countries like Italy or France.
Yes. So you'll be amazed to know that when we first wrote the app, it was available in 15 languages. Sorry?
Really?
And we used it on a research basis. So it's actually being used currently in South Korea, in Seoul, in Hong Kong. It's being used in Tübingen in Italy, Marburg in Germany. It's being used in Montpellier in France. So we've got it in all those languages. But that's through a research contract. So that means these are people where we've got a research agreement, the app is being made there, and it's not feeding back to the patient. The new health app, I helped to set that up, that company, because I got frustrated with not being able to make it available to more patients. And so now what they've done is taken it that bit further. The app has also got cognitive memory tasks. It allows you to program it to remind you when to take your tablets. You can actually say whether you're on or off when you do the recording, and you can rank your non-motor and your motor symptoms. And if your non-motor problem is constipation, it's going to link you to how to self-manage that first. So what are things that you can be doing as a patient proactively without having to kind of like ring up your Parkinson nurse or see your consultant? So it's really about more ownership, putting the care back onto the individual through empowering you and learning more, but at the same time about the person who's looking after you having access to that data and trying to use that to inform on, treatment decisions. I've now forgotten what the question. Oh yeah. So at the moment, I'm aware that the app is being tested for free in 6 NHS places. I think we're likely to we've got 2 large papers currently being worked up to be submitted in the next month or 2. So if they get published, I think more people will be interested in using it. I know that the app now also has investment from the US in Cedars-Sinai. So that means it might be going over to be used in America. And ideally, you could make it available, what I would like, at a very reduced rate for somebody with Parkinson's on an individual basis to trial it. And you could be getting the money for all the investment and the development by using it in drug trials or insurance companies, for example. So, that's kind of how I would hope the market will go. So I'll tell it very quickly. When we set it up, literally we got tens of thousands of smart recordings from local people with Parkinson's in a matter of a year.
Wow.
You've got your hand up, Barry, or did you not?
Yeah. Just a few things. Thank you very much. just have to agree with Mary. Exercise and mood are just so powerfully effective for me. I actually went into my last appointment with the nurse at the Parkinson's clinic, and she looked at me and she said, "Barry, what are you doing?" And I thought, "What have I done wrong?" And she said, "You seem to be reversing your condition of Parkinson's." It was quite spectacular. And it's a lot of it's through state of mind. And I just feel I'm in the best mood that I remember myself having been in since I was 40, and I'm 67 next week. And I just seem to get better. I really feel there's something that one can work on that can make you feel that Parkinson's can be a regenerative as much as a degenerative condition. And it's through the mind, it's through the state of being and being active.
Yeah. Let's say also that your diagnostic experience was a positive one because you saw a nice neurologist who was interested in Parkinson's and told you all about your treatment and how you can proactively manage. So you were educated, you were given a lot of information, you took part in research. You are more likely to be motivated then to do things like exercise. And, by exercising, you're releasing endorphins, you're releasing serotonin, dopamine. All of these things are going to have a beneficial effect on your mood, your sense of well-being. And, I have many people with Parkinson's who tell me that the other beneficial effect on their tremor or their motor symptoms can last for 24 hours after, or even longer for some people.
And Mary, just to say, I cramp in my one foot, and sometimes the toes go in and sometimes the toes go out. It's quite peculiar. But it's almost exactly between 6 and 6:30 in the morning. And if I take through the night a slow release dopamine tablet, Carbidopa, and I'll never take the second one later than 5 in the morning. I have a funny waking up regime. But if I take one in the later part of my sleep in the night, then I don't cramp. It's quite clear about that. It does have an effect.
Okay, can I just underline one of the things that European Parkinson Therapy Centre does teach everyone is that you can affect your condition. You can take control of your condition.
Yeah.
You can change your outcome to a certain extent. Even Bas Bloom was quoted recently as saying exercise was the only thing that actually worked in slowing down the progression.
Well, that's correct based on the evidence we have so far.
It is indeed. Okay. Any last questions? Well, I would like to thank Michele very much. There's a couple of questions in the chat. Should women be given a smaller dose of drugs in trials?
This is really important, actually. I'm glad somebody raised that, because what we found in our research is that women are lighter in general than men, but they're not given the same. They're given the same dose as men, and therefore, on average, they're more likely to be overdosed than underdosed, and they run into these dyskinetic movements earlier. And the other thing for women is not really recognizing the effect of menopausal symptoms, HRT replacement, on how your Parkinson's manifests. So things like a frozen shoulder seems to be very common in women I look after. Really bad joint pains and tendonitis, which actually often improves with HRT and Parkinson's treatment and diagnosis. So I think that women are an under-recognized group in Parkinson's. They don't get it as commonly, but they're also under-recognized in terms of research. So they don't participate in to represent women. The participation is under that what it should be. And this is something that research also needs to address, as well as having more ethnically diverse participants from many different cultures and countries, because the vast majority are in more wealthy countries like the UK and white European, Northern European stock.
Okay. We're pretty much on the hour mark, and I promised I would keep you only for an hour so you can get some dinner. I'd like to thank everyone who's joined in. if there's any last words, and I don't mean last words, I mean last words. There's some thank yous coming through the chat. That's excellent. So we're very grateful to Michele for giving her time and courage, and for the information you gave to us, Michele. You'll always be a star in European Parkinson Therapy Centre. You've always been there for us. So,
Alex, you are the star through what you've set up and done and demonstrated, actually. And you know, don't underestimate what you can do and the effect that the centre has had. I mean, I never stop recommending it to my patients. I've had so many people go and just come back after a week, and they just said the effect, the benefit's been tremendous. So just also to plug the PD European Therapy Centre as a really good way to just sort of, you know, take stock of your Parkinson's, and also to have a lovely Italian holiday in the Alps with your partner who can eat pasta, stay in a pensione, and enjoy fun things with you too. So
I am doing just that this year.
Highly recommend it, Mary.
Very much.
I visited the centre. It's really wonderful.
I think I will enjoy it and benefit from it very much. Thank you.
You will,
And
Mary.
I agree entirely with. Can I get your app here in Ireland? Can I get the app?
Yeah, so we are working on it. Just give us a little bit of time, because what we're trying to do is collect the evidence to persuade NHS trusts you know, that it's worth doing, and also to expand it so it's available to people at a very low cost, right? So that's what I'm working on. Yeah.
That's very good. Thank you very much.
Pleasure.
Thank you.
We would love to have a copy of the app over here. We'll talk about that.
Yeah.
Anyhow, if I could just say goodbye and good night to you in particular, and thank you for the time and the energy and the preparation you put in. It's not always that easy to talk off the cuff, but you've done a brilliant job, Michele. Thank you so much, and good night. Good night, everyone.
Thank you.
Bye-bye. Thank you.
Bye.
Bye.
See you next time. God bless.
It was.
Like a phoenix rising from ashes of doubt, you'll find your wings and soar about.